What if I told you that right now there are 10 times more bacterial cells than human cells inside of you? Before you freak out, just know that a lot of these bacteria don't actually harm you at all. Some nasty ones, however, can cause infections. Fortunately for us, humans have developed antibiotics to fight these infections. Over the years, however, more of our antibiotics are becoming less effective. In fact, by 2050, it is estimated that more people will die from antimicrobial resistant bacteria than cancer. Now, could this antimicrobial resistance be due to the ever-growing population? Is it our diet, or is it the combination of drugs we take? Let's find out. It all starts with Darwin's theory of natural selection, which states that organisms, including bacteria, can undergo random mutations. Although many mutations are useless, some actually increase the bacteria's survival by developing resistance to a certain antibiotic. So how exactly do these bacteria develop resistance? There are four main ways in which bacteria can develop antibiotic resistance. The first being antibiotic modification, where the bacteria preserves the same target as antibiotic-sensitive strains, but the antibiotic is prevented from reaching it. This happens with beta-lactamases, where beta-lactam ring is cleaved, thereby inactivating the antibiotic. The second method is by preventing the antibiotic from entering the cell, or by pumping it out faster than it can flow in. The third method is by altering the primary site of action. Because of the structural change, the antibiotic can penetrate the cell, but isn't able to inhibit the activity of the bacteria. And the final mechanism is by producing an alternative target, which is usually an enzyme, whilst continuing to produce the original sensitive target. This allows the enzyme to bypass the effect of the antibiotic. The best known example is the alternative penicillin binding protein, PBP2A, which is produced in addition to the normal penicillin binding proteins by MRSA. The protein is encoded by the MEK-A gene, and because PBP2A is not inhibited by antibiotics such as flucloxacillin, the cell continues to synthesize peptidoglycan and hence has a structurally sound cell wall. There are two types of antibiotic resistance, inherent and acquired. Acquired resistance occurs when naturally susceptible bacteria gain the genes encoding resistance mechanism via mutation or genetic material being transferred from another bacteria. Now, transfer can occur via three ways. Firstly, conjugation. This happens when direct cell-to-cell contact with plasmid transfer. Transduction. Transfer of bacterial DNA by a bacteriophage. And transformation, which is taking in naked DNA from the environment. Antibiotic resistance has led to the rise of the so-called superbugs. The most dangerous of these are the bacteria that include Neisseria gonorrhoeae, Staphylococcus aureus, Streptococcus pneumoniae, and Mycobacterium tuberculosis. However, we will focus on the two that are considered most deadly. Staphylococcus aureus is the number one cause of hospital-associated infections, with invasive infections having a 20% mortality rate in the hospital. In the hospital, golden staph makes its way to open wounds or via invasive medical devices. In just 72 hours, MRSA can progress from red bumps to pus-filled boils. Death is caused by sepsis, attack of vital organs, and necrotizing pneumonia. This bacteria produces an alternative penicillin binding protein that diverts the antibiotic away from its cell wall, producing enzyme, leading to the rise of methicillin and vancomycin resistant strains. Tuberculosis, or TB, is one of the leading causes of death worldwide. In 2013, 1.5 million died from it. It is caused by the bacteria Mycobacteria tuberculosis, and it is an ancient organism, with its favorite host being the human. TB is transmitted through the air and forms granulomas in the lungs. The tuberculosis can then spread via bloodstream to form other tubercles throughout the body. If left untreated, the chance of death is 66%. Tuberculosis is now resistant to multiple drugs. Mechanisms of resistance include spontaneous mutations that modify drug-binding targets and efflux pumps that expel multiple antibiotics out of the bacteria as soon as they enter. So what can we do? We can start by stopping antibiotics used in agriculture, and the education of prescribers to not prescribe for minor illnesses, and the stopping of over-the-counter antibiotics, especially in developing countries. Now, infection control includes hand hygiene, isolation, and screening. Novel methods to fight superbugs include using viral bacteriophages, lysins, radioimmunotherapy, vaccines, and ways such as iChip to identify or discover previously undetectable pathogens. In summary, we can see that bacteria are all around us and in us, and have developed resistance to the antibiotics we use to get rid of them. With above protective measures and other research developments, we hope that one day bacteria can resist no more.